Good, good, yes. There are some videos I posted on Blackboard. They're in the recorded lectures section. I'm double checking myself on my other screen just to make sure. Under the class lectures folder, you know where you normally find all of our lectures, there's a folder called Crash Course. Yes, Crash Course Biology. And it links to a bunch of YouTube videos. If you click on the folder, you'll see all the ones I'm talking about. And Beth sent these to me. She sent them to the rest of the class too, which was really nice. So I appreciate that. But there's a fella on there going over the same information in the chapters more or less than I am. I haven't had a chance to watch them all yet. Seems to me they're no longer than about 15, 20 minutes a piece. And again, I haven't had a chance to really dig into them too much as far as that goes. But the little bit I have seen of them, I like this guy's style. I like this guy's energy. As I mentioned at the beginning of the semester, with the internet and your access to it, you got this great opportunity that if I just went over the lecture on, I don't know, meiosis, right? And you're like, man, I tell you, I really didn't get that crossing over part, what he meant by that and how that was working. I guarantee you go on YouTube and you type in meiosis lecture, crossing over lecture, or something like that. There's some YouTube video. You might even find one of my old ones up there from a previous class, but there is somebody up there talking about that, probably doing a much better job than I am, especially with animations and that kind of stuff. So even though I do try to post some videos for you, these are just ones I find. There are literally hundreds of thousands, and that's why I appreciate when you all find cool stuff that you send it to me so that I can make it available to everybody. So, oh, also, by the way, speaking of videos, I've done some videos on meiosis and crossing over, and one on those genetics problems that we started the other day. There's a video from a few semesters ago. I've posted those under the topics and concepts part. I sent y'all an announcement. So click on the link on the announcement. It'll take you to where those are. Now, it's an older version of Collaborate. So you have to, first time you load it, it's going to ask, can it install this thing called Windows Launcher? Yes. And then just follow through the prompts and accept everything. Everything's cool as far as that goes. And eventually it'll load the older Collaborate. I haven't, yeah, exactly. That's what I like about, being able to do it. My videos here, my lectures here are much more what you see in class, obviously. But this guy does a great job of bringing in some technology and using that to illustrate stuff. That's really always the challenge, isn't it? So anyway, take a look at those videos. If you have problems loading the older Collaborate, let me know. I haven't changed them over to the newer version yet because we're actually getting this ready to switch over to a completely new something called Canvas instead of Blackboard. Okay, so if there aren't any questions, I do want to get into lecture because time is a ticking, right? Just to kind of step back for a second, I did want to show you something as I was flipping through the slides this morning. I reminded myself that, oh yeah, if you look at the difference between being a purple flower or a white flower, if you look at the different, Mendel's flowers, if you look at the difference in the phenotype, you'll notice that how we've talked about changes in genetic information can sometimes have bad effects or no effects, and sometimes just like sickle cell anemia, right? Those kinds of changes sometimes can have big effects. Look at the only difference in the DNA between the gene that codes for purple flowers and the one that, basically, if you look, the one that codes for the absence of purple. You see up above, you notice that the gene, if it's expressed, is going to help the flower synthesize purple color, and that's why it's purple. But if you don't have the gene that makes that enzyme, it's an enzyme that does that, and the only difference, that first base, look, up here it's a cytosine. Now don't worry about which side is red, is it the cytosine or the guanine? We'll talk about that later on. And down here it's just one base, an adenine. Isn't that neat? One little change. Okay, so and if you think about that, when we look at genetic information, remember that genetic information is typically very complicated, right? With a series of genes controlling physiological reactions. There's lots of little levers that can be flipped, right? Is it this letter? Is it this letter? You got 4 different letters you can choose from, right? The guanine, the cytosine, and the A and the T. So lots of variation. Of course, I've said that word a million times, even to you guys, right? Variation. Know these ratios, okay? Because that's the predictability of science. It's one of the things we want you to learn. in our science courses is the understanding of analyzing data and coming up with reasonable results. Know these ratios, right? So if someone was to say to you, and I'm going to be very general, trait A is dominant, whatever trait A is. I'm not going to, flower color, whatever. I'm just talking in general. If trait A is dominant, okay, and we're going to indicate the same trait as recessive with the lowercase, right, sticking with the letters that we use, you got to be able to tell me that if I cross these two types of critters together, I'm over on the right now, heterozygous. If both the parents are heterozygous, you got to be able to tell me how many of those are going to have the dominant phenotype, excuse me, in other words, in this case, be purple or whatever that trait might be, and how many are going to be recessive, right? And that's what that 3-to-1 phenotypic ratio tells you. 3/4 of them will have whatever the dominant phenotype is. When we say the word dominant phenotype, what we mean is the phenotype that you express, this case purple, right? The phenotype you express if you have at least one dominant allele. Remember, you don't have to have both. Because remember, if you look at genotypic ratio, only 1/4 of them are going to be homozygous dominant. Half of them will be this way. And then, of course, there is the white ones, right? Okay, so make sure you understand and know those ratios, because that helps, like I said, that's the point, predictability. I might ask you questions on exams, like we did with the fruit flies, about how many are going to be this way and how many are going to be that way, and so on. So, guy did this with an artist brush. Isn't that amazing? So he would go and he would, maybe the first part isn't so nice to the flower, but since they don't have nerves and brains, it doesn't matter. He would go it off, and what he's cutting off is the stamen, which are the male parts. And then he's using the stamen, like you see down in number 2, he's using the stamen from the white flowers, the male parts, to fertilize the carpel, which is the female parts. It's interesting, I read an article the other day that in China, they've lost a lot of their pollinating honeybees. Honeybees are really good, pollinators, going from flower to flower. They scatter pollen, which is necessary for plants to grow, to propagate, right? But because of pollution and environmental destruction, they're pollinating honeybees, and not all bees are pollinators, why I keep saying pollinating honeybees. Or their pollinating bees population has dropped, like almost to nothing. So one thing China does have a lot of is with people, right? So what they've done is they've actually got folks out in the fields with paintbrushes going between the male parts of the flowers of whatever crop they're growing, and then spreading that pollen to the carpel. So they're acting as the bees. Can you imagine how exciting that job would be? Maybe for a couple of days, right? For me, it'd be like, oh man, check out. But I mean, after a couple of thousand flowers, right? Excuse me, I got a little bit of voice cracking getting toward the end of the end of y'all semester. All right, so this is, of course, what we talked about the other day. And if I went down below and I said, all right, folks, look at these numbers. Oh gosh. Well, I hope that the recording is better. It's not just something that's going on because I can't tell with information coming in is being broken up. When I look at your signal strength, it says it's great. So let me know if the recording is messed up, okay? All right, so if I went down below and I said, okay, tell me what ratio you think that is. 705 to 224 white flowers, 705 purple. What kind of ratio would you expect that would be? What would that simplify to? There you go. There you go. Oh. See mine? No, mine's running okay, so I don't. Could be the school too, because this all goes through the school server. So like I said, I hope the recording is all right. So again, table 14.1 shows you that. Oh, is that right? That could mess things up. So table 14.1 is just sort of a table of different pea plant traits, right? Remember, we did with flower, but it doesn't matter as long as you know which one is dominant and recessive, which, like I said, I'll tell you. Some other genetic traits that Mendel looked at, where he looked at the color of the seeds, right? Yellow and green. Yellow is obviously dominant here, right? He looked at seed shape. The round was dominant. Look at the ratios over on the right. Remember, they don't come out exactly 3-to-1. They may come out 76%, 24%. You get the idea. But Notice that no matter what trait we're looking at, the dominant trait to the recessive trait ratio is always about a 3-to-1. So if we looked at flower position, axial, and terminal, you can see what it means. Flowers that are terminal grow at the end of the branches. Flowers that are axial grow down the branches. You get the idea. We're not botany here. All right, so no matter what trait we looked at, that 3-to-1 ratio, and it's not just in pea plants, folks. In human beings, traits that are inherited this way, and now, not all traits are inherited simply in human beings. What I mean by simple is we're only doing monohybrid right now. We're only looking at one trait. So, for example, a pea plant with, what do we say, green pea pods would be the same in our numbers, in our counting, if it were tall or short or dwarf, right? Of course, those are 2 different traits. There were, what, 2 on the other page, 3 on the other page? So there's 7 traits here. Human being have, 30,000 different traits, right? So when you put all that together, it becomes much more complicated when we start adding more traits. So this whole idea of what's going on and the reason why we have to do Punnett squares is because, like I said, Mendel understood probability. He understood that which sperm does the job, which sperm does the fertilizing, which egg gets ovulated. Now, all that sort of stuff that we talked about was just like tossing a penny, right? 50% of the time, you're going to get sperm with this allele, and 50% of the time, you're going to get sperm with this allele, right? If you only get 2 alleles, purple and white, like in the flowers, your sperm are going to be half purple and half white, unless there's some other weird thing going on genetically, which would cause the ratios to shift. And there probably is, but nothing comes to mind off the top of my head, right? So we're just going to play the probability here. And that's why he gets the ratios down below. This whole idea that, this whole realization, is that the word I'm searching for? This whole conclusion by Mendel that we have to use a Punnett square, that we have to count for all the ways that things could happen before we can make predictions, right? Down in the Punnett square down below, we have to imagine that, well, maybe a dominant alleled sperm fertilizes a dominant alleled egg, but it could also fertilize an egg with the recessive allele, the little p. So all of those combinations we have to account for, and that's why we come up with ratios. So Mendel's not going to say in that particular cross 100% of the flowers are going to be purple. You say about 75%. Okay. Now, there is one place when he could say to his betting monk friends, every flower that's going to be born is purple. There is one cross he could do. There's 2 flowers he could cross together and always be certain that he's going to get purple flowers. There's also 2 flowers he could pick, 2 colors, or 2 flowers. Could be of the same color, don't let me mislead you, that he could 100% of the time predict the offspring, couldn't he? Does anybody know which ones I'm talking about? Homozygous dominant and a heterozygote. So let's, everybody agree with Tim? 100% of the time, Mendel says flowers are going to be what? What color? Right. Or even a puppy. Okay, now, how about the other way around? Is there another cross that we can do? Homozygous, yeah, there's another one. That'll make nothing but purple. There you go. One more. There might be more, but one I'm thinking about. What's another cross we could do that you're 100% guaranteed to get the color of the flower? Yeah, cross 2 white ones, okay? Now you're understanding what I'm saying, right? But if we don't know what sperm is going to fertilize what egg, and we have no control over that, we have to play the probability game. Makes sense, right? Now, there is a couple things we can do to give us information. Like if we don't know if that purple flower or that dominant phenotype is true breeding or not. Now, let me define true breeding again so when you're reading it someplace you don't get confused. There are 2 ways to be true breeding. True breeding just means you're homozygous, that's all. Right? So you can be homozygous dominant or homozygous recessive, and your true breeding. What Mendel was testing in a test cross, a monohybrid test cross, was the purple one true breeding. Okay, see, the reason why you don't often see true breeding and white put together, it's kind of redundant, right? It's kind of like saying a round circle. Because you can only be white, you can only be true breeding if you are a white flower. Get what I mean? But by definition, so that if you see it, you're not confused, true breeding just means homozygous. Okay, and this is the test cross. And we talked about this in class, and he was able to predict the ratios like Tim said earlier. If you have a homozygous dominant or heterozygote, a white one, you're going to get all purple. But on the other hand, if you get some of the sperm have the white allele, like in the situation on the right, then you get a 50-50 offspring. So you could say, all right, well, if this flower I picked is not true breeding, so he's saying if this flower I picked is purple, but it's heterozygote, I'm going to tell you half will be purple and half will be white. Okay, you ready to go next level? That's one trait. Now, this model of genetic inheritance, Mendel, works for not all genetic traits and not all cases, but generally it does. So we're going to add another trait. We're going to now talk about dihybrid crosses. All right, and we're not going to go any further than that, but you can, is what I'm trying to get across. There are obviously trihybrid and there are obviously as many crosses as two traits, or as two parents bring traits to the table. In the case of human beings, what did I say? What is it, 30,000 genes? I don't know if that's 30,000 traits. Many genes sometimes control a trait. But let's say several thousand genes in the male, several thousand genes, equal number. Well, maybe not necessarily. But anyway, in the female, you mix that all together and you get this multi-hybrid cross, right? That's why we get all this variation. So what's going on still works, right? But it just works at a much higher level. So let's do that. Just go to the next level, dihybrid. Okay? All right, now in dihybrid inheritance, what we're going to do, many generic parts. Are there many generic parts between us? Who's us? First, Tim, you talking about males and females, or animals or humans and animals, or is there a lot of genetic difference between us humans? Is that what generic parts? If you mean generic parts, are there tissues that aren't specific to me that everyone else has exactly? Oh gosh, well, there are tissues. Yeah, we all have digestive tissue. We all have nervous tissue. How that digestive tissue does its job is going to be confined to a range of efficiency. All right, so I'm kind of getting into evolution here. In other words, if you had a very inefficient digestive system, that wouldn't be passed on evolutionarily. But of course, each one is going to have a certain amount of variability that's allowed within the constraints of still being able to do the right job. Does that make sense? So while your genetic information in your nervous tissue might be a little bit different than the genetic information in Raymond's nervous tissue, if they both get the job done, right? Then, and maybe this is where we look at variation. Maybe in Raymond's nervous tissue, there tends to be some genetic information that gives him a little better computational, awareness, right? So he's a little better math. Maybe that explains it. It's not saying that those kinds of things can't be overcome. That's why I always object to, well, you're just not good at something like that. I think you can overcome that. It's the fact that maybe you pick it up faster. Like, I wasn't really good at math, but I picked it up because I studied a lot. Some folks, like a buddy of mine, sat down and it was like nothing to him. Yeah, Tim. Yeah, you got it. If someone was born, excuse me, with genetic information in their nervous tissue that was way outside the norm, like just some genetic mutations occurred and they made all these really different kinds of proteins, and they thought in this really different way, that you're probably going to get something that's not going to work. So that individual would probably not be born or something like that. You might get something like the Einstein dude, who could look at a clock as he drove away. That's how he figured out this time stuff, folks. He's sitting in a carriage, driving away from a clock in some place in Switzerland, going down the road, and looks back at the clock, and he goes, the further I get away from the clock, the longer it takes the light from the clock to get to my eye. Therefore, with his genetic information, maybe, he figured out that means time changes. You can stretch time, okay? And then most of us are going to be in that mental range in the middle. Okay, do you see what I mean? It's like a big bell-shaped curve. Good questions. Yeah. So, you know, it's good that you guys are thinking in those ways of biology and how that relates to evolution and how that relates to diversity, because that's what you're doing by your questions. You're understanding that while diversity is, you know, occurs among all of us, that Mother Nature, you know, along those biological realms says, okay, but you got to keep it within, unless you come up with something really good. Unless you come up with an Einstein, then that's cool. But, you know, you got to sort of keep it in the boundaries there. Oh, yeah, or bad, yeah. And y'all need to take my Bio 2 class. We'll talk about that for about a month. All right, let me take a drink of water. So what are the 2 traits that we're looking at here? Could be. I don't know. You know what I always like to think about it is your intellect and your, let's say your academic ability, or efficiency, whatever you want to call it, and your mental stability. It's like one blanket trying to cover a bigger blanket beneath it. That's why so many times folks that are really smart are just kind of odd, right? Blonde hair, blue eyes, wing length. Those things are connected, linked. I'll talk about that in a second. But that's why so many times when you see someone with blonde hair and blue eyes, and it works in fruit flies too, eye color and eye wing, you go, wow, why are so often blonde-haired folks blue-eyed or blue-eyed folks blonde-eyed? Is because those genes are close to one another. So let me yeah, I think so. And artistic creativeness, if you think about some of the artists, right? They're always, you know, which is great. They think differently. Okay, okay, the 2 traits we're looking at here are going to be the color of the seed, yellow and green, and the shape, which we're going to call round and wrinkled. And Mendel's going to start off just like he did with the purple and the white experiment. He's going to start off with true breeding, yellow round and green wrinkled seeds for reasons that you understand. Okay, the test cross, right? That's how he's able to figure that out. Now, if you go down below, and what we've got down below is we say, all right, what kind of gametes can each produce? And he circles the gametes in the illustration just like I do. So it's and probability. You got to figure out all the types of gametes each parent can produce. But in this case, just like white flowers and white flowers making white flowers, there really is only one probability, right? If you're a homozygous dominant, now, here's how we're going to change the terminology a little bit in dihybrid. I'm going to use the word like, say, doubly. Doubly homozygous dominant, doubly, meaning both traits are homozygous dominant. So if you're doubly homozygous dominant, the yellow round one, and you're doubly recessive or doubly homozygous recessive, and the green and wrinkled one, then you're only going to make gametes that have the dominant alleles or the recessive alleles, depending on which one you are, right? Make sense? Yeah, it's you know, if you only have 2 numbers to, one number to choose from, then it's pretty obvious which number you're going to choose. Okay, choose a number between 1 and 1. Well, I can predict what number you're gonna choose. All right, now it gets a little more complicated when we cross the F1s. Same thing he did with the purple flowers, right? I said, we might even be thinking about Mendel if he hadn't done that, but what he does is he does this. Takes another yellow round one and he crosses them. All right, now things get a little bit wonky, right? Things get a little bit funky there because there's many different ways. Well, not many, but several ways that those gametes can come together. You know, for example, you could make a sperm or an egg, depending on your sex, that has the dominant allele from the first trait and the recessive allele from the second trait. Now, I'm circling that again as a gamete, and you're going, wait a minute, he told us gametes have 1 letter. Gametes have 1 letter per trait. With the flowers, we were talking about just color at the time, right? So there was 1 letter. Now we're talking dihybrid, so we got 2 traits. So I have to have 1 letter per trait. And in a gamete, there only should be 1 letter per trait. There should not be 2 Ys. Should not be 2 Rs. There should be a Y and an R, okay? And you should have either the dominant or the recessive in those. So you can easily go, gosh, how do I remember all of those alleles? Well, you already know how to do it. Let me get a blank piece of paper. Let me get a blank thing here. Where's my mouse? There we go. Hold on 1 second, because I want to write this out. This is Back in math, those of you going through math, you learned a little mnemonic device. You know, I love those things, by which you remembered the order by which to do this. Do you recall what that is? Those of you who have, do you remember that little mnemonic device? How do I expand a binomial? That's what that's called. Maybe you did in college algebra? They must teach that in college algebra. Maybe intermediate algebra? Yeah? Do you remember that little mnemonic device, the little 4-letter thing that remembers that you multiply the first ones together, and then you multiply the outer ones, and then the inner ones, and you add those together, then you do the last ones? Do you remember all that? Oh, there you go. Thank you, Raymond. FOIL, first, outer, inner, last. Okay, now, you know how to do that, right? Other than the adding of the outer and the inner, you're going to use the same idea to figure out those gametes. So let me go back to that. So we got this kind of thing. We got a doubly heterozygous. In order to make sure that you get all the possibility gametes that are possible, in order that you record all the possible ways that gametes can be formed. Now, remember, if you forget a way, that's going to change your numbers. If you were flipping a penny, to make it really simple, and you didn't realize that there was a tail, and if you said, okay, I'm looking at this penny, 100% of the times, if I toss it up in the air, it's going to come up with heads. You didn't account for the fact there's another side. Okay, you wouldn't do that, but in genetics like this, and in statistics, sometimes that happens. So here, let's use this mnemonic device. Okay, what that says first. First says that one of the gametes can have the first allele from each trait. Right? This just happens to be the dominant ones. All right, so I go up and I like to check these off. First ones are done. First, done. Outer. All right, now you're not going to add anything, but you can see what I mean. Outer is going to be these alleles, the ones that are on the outer of the genotype, because that's what that is. That's the genotype. So I got the. I shouldn't put checks there. Anyway, outer, inner, these guys. And then last will be the last alleles. First, outer, inner, last. We're done. That's it. That's how many they can make. That's how many different ones they can make. Now, again, before I get off of this and go back onto the screen, look at those gametes, sperm or egg, right? And are they genetically different than one another? That's the whole point of meiosis. Are they all a little different than one another? Or are there any repeats at all? Anybody see any repeats? No, they're all different, right? They're all different. So now what you would do, right, is you would take all of those different types of gametes, and that would be the ones that one parent can produce, and you would multiply, or you cross, not multiply really, with the same setup for the other parent, right? Let me try to write these real quick so there we go. And right there you go, whoa. So I mean, I got to figure out all the possible ways these can come together, and then these can come together, and of course that one could also fertilize that one or be fertilized by that one. So you got this one there. Before you know it, you've lost track and you're going to lose the game of prediction. You're not going to be able to figure out the ratios because you're like, oh man, I forgot about putting those two together. So what are you going to use? Your good old buddy, the Punnett square. Now it really becomes obvious why you need one of those things, doesn't it? Now you say, wow, if I just have some visual tool to figure all this out, I'll make sure I not miss anything. Okay, you ready to go look at the Punnett square now? Be much less intimidating now we go back to it. So let me go get that. So anytime I ask you to do those dihybrid crosses, remember FOIL to figure out the gametes. Now, you might end up finding out that half the gametes are one way and half the gametes are the other way. You only have to put one of those into the Punnett square if they're the same. And I'll show you what I mean when we start doing some of those genetics problems, because we still have to do the dihybrid ones. 14. Get this loaded. All right, we were dihybrid cross. 12. Yeah, my computer is kind of hitching too, so. Okay, there we go. All right, so the Punnett square that I'm talking about is the one on the right. The hypothesis of independent assortment. Okay, what that means, and I put it in red, is that that's another one of Mendel's laws. Mendel came up with a couple of laws. The one was the law of segregation, which was telling you that what happens is you have to account for the fact that alleles have an equal probability of ending up in gametes. So in other words, if you're heterozygous, half of your sperm will be the dominant allele, half will be the recessive. Now, that doesn't apply for homozygous dominant or recessive, but we're talking about the heterozygotes. In the law of independent assortment, what Mendel said is the trait for yellow is going to segregate off, just like we talked about with the purple alleles, and go off in a gamete by itself, independent of what the trait for seed shape does. So in other words, seed color and seed shape are not related to one another. So they're going to sort out as if you were just looking at them by themselves. That's why you get a much more complicated table. You get a 4x4. See that Punnett square on the right-hand side? And all they've done is from that scratching that I did on the sheet of paper is I've transferred over the different kinds of sperm and egg. Now you notice in this case they got a little 1/4, because you remember before it was half of the sperm will have the dominant allele, half will have the recessive. But now this is a half times a half because you got two traits. Okay, so that means there's a 25% probability that your sperm will have the dominant allele from both traits. 25% you'll have the dominant from one, recessive from another. Oh, it's there. The R is there. It's just somehow mistyped below. See where it's kind of hanging out in that box? Should move that up next to that Y. Okay. All right, so you get the idea. Same thing for the eggs. And then in the body of the table, what we do is we fill in the crosses. We say, okay, what if this sperm fertilizes this egg, blah, 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 blah. And we run through, and then we count up the phenotypes. Okay, now this becomes a little bit tricky. There is a genotypic ratio in dihybrid crosses, just like there's a genotypic ratio in monohybrid crosses. But I'm not going to bother to have you guys know that, to memorize that, right? 1/16 them are doubly homozygous, 2/16 are homozygous for the first, and forget that. Let's just worry about a phenotypic ratio, because that's what we're really looking at anyway. What color are the seeds and what shape are they? So you understand there is a genotypic ratio, but for the dihybrid, just because it's really sort of doesn't give us a lot of information, let's really focus on the phenotypic. And notice it's a 9-3-3-1. So that means 9/16 of the offspring are going to be yellow and round, and then 3/16 will have one dominant trait and one recessive. So they'll either be green and round or wrinkled and yellow. And then a few of them, just a 1/16 of them, are going to be doubly homozygous recessive. So they're going to be green and wrinkled. It's always going to be a 9-3-3-1. Is it always going to be exactly a 9-3-3-1? No. Look down below, and there's some of the data from Mendel's work. 315 yellow rounds, 108 green rounds, etc. That doesn't come out to exactly 9-3-3-1, but it's pretty close, just like the 3-to-1 ratio. Okay, now what I would do, and your genetics problems will help you do this, as I would practice doing some of these dihybrid crosses. I would cross parents that are doubly heterozygous. Well, you can cross any of them, but I would definitely make sure that I cross parents that are doubly heterozygous and make sure that I get those ratios that I should get. Pick any trait you want. There's a few of those in the genetics problems too, okay? Now, what you're looking at on the left-hand side is basically him doing a test cross, okay? And in this particular case, his test cross came in with a 3-to-1 phenotypic ratio, but he didn't get that particular ratio. That's because he discounted this hypothesis. In other words, he said these alleles for these traits, yellow, color and seed shape, will end up in gametes independent of one another. Remember I said that? But he had an alternative hypothesis. It's science, folks. If it's this way, this is the results I get. If it's this way, because I come up with two reasonable hypotheses, these are the results I get. I didn't get these results. I got these results. Not only do these results support independent assortment, these results refute it. These results also support it. It wasn't that way, it was the way on the right. Folks, that's the way science works. Now, so practice those. Down back to Raymond's original question, linked genes. Not all genes that seem to be that they should be independent sort this way. In other words, you get different ratios. Like, for example, when you see blonde hair, we often see blue eyes in humans, don't we? Right? Blonde hair, blue eyes. Not always, but isn't that a very common trait to inherit together? Now, you don't know anything about fruit flies, but among them, having a certain eye color and a wing length also works the same way. In other words, these traits that seem, they're two different traits: color of your hair and color of your eyes, and then in the fruit flies, the color of your eyes and your wing length. But for some reason, they don't sort out 9331. That's because they're linked. Now, what we mean by that is they're very close to each other on the chromosome. They're what we call linked genes. And genetic information that is close to each other on a chromosome, very often during a chiasma, right? During a crossing over, very often they get inherited together because they're so close to one another. So is Mendel's law of independent assortment wrong? No, it's usually always right. But one thing you have to love about science, and I've always loved about biology, is not everything is perfectly right all the time. There are those, and you can imagine how this would confound scientists at the beginning, right? They're going through going, I got it, and Mendel and these guys after him, I got it, it's 9331. You want to bet? 9/16 of them will be A and B, right? And then all of a sudden someone says, okay, what about blonde hair, blue eyes? He makes the bet, he loses. Can you imagine? Because we didn't realize at the time, of course, that these genes are linked. That's what we mean. It's physical. They're simply close to each other. So when those crossing over points occur, because they're close to one another, they very often come as a group. Remember, those crossing over points are random. Makes sense? That's what we mean by linked genes. There are lots of linked characteristics in human beings. You look it up online. There's one called Marfan syndrome, which is actually a disease, but it's the same kind of idea. It's a bunch of linked genes. All right. What was it? Lindsay the other day said something in class about flowers, right? Didn't you, Lindsay? The red and the, how do we get pink, didn't you? Isn't that you? Yeah. All right. Well, I'm getting ready to tell you. You see, most of the time, the way these traits work is they work just like we said. Dominant, recessive. Dominant is dominant over recessive. In other words, dominant expresses itself. Recessive doesn't express itself unless, of course, there is no dominant, right? That's why the green wrinkled seed is green and wrinkled. You notice for the color, there's only recessives, and for shape, there's only recessives. It's the same thing as with the white flowers. But the question you brought the other day was, well, how do we get pink ones? Well, some of the very early work on genetics was done with things like flowers. Mendel wasn't the only one. Lots of other folks were working on genetics, and they were using flowers. They're easy. Easy. You got to get a paintbrush and do it, but they're easy to figure out the traits you're looking at. You can decide who breeds with who, right? You can say, all right, let's test this purple one with this white one. So it was done with flowers. But flowers had something up their sleeve. It helped us understand a lot of things about biology and genetics and organisms in general. But I think at the beginning it had to be pretty confusing. All right, let's talk about this in typical Mendelian terms that we've learned so far. The red allele, in this particular case, the red allele is being indicated with a C, capital R. The C usually indicates there's some chemical that causes it to be the dominant allele. But let's just for a second say that the red allele is dominant over the white allele. Right? That's what we've been saying all along. So if you look down at the F1 generation, don't look at the results, but based upon that assumption that the red allele is dominant, not incomplete, just dominant in the terms we've used before, what color should all of the flowers have been in the F1 generation? If that was your hypothesis, is the red allele dominant over the white in, what are these, snapdragons or morning glories or something like that? All of them, every single one of them? So you had 1,000, should all 1,000 of them have been red? No, you're right. You're right. Look at the cross up there. All you've got is red and white allele. See the gametes down below? So if we're talking with, if this was red was dominant, this is like purple and white. They should all be red. They should all be purple. But they weren't, you see. You weren't, in spite of the fact that, in spite of the fact that we We saw that in the pea plants, and it seemed to work in many other traits. Once again, Mother Nature loves throwing us curveballs, and that's, again, some of the excitement of all this. Not everything's the same, because what happens in this is the red is not dominant, you see. It's not dominant. It's incompletely dominant. So what does that mean? It means it's not completely dominant. That means that a little bit of the red information is there, maybe some enzyme that synthesizes the red, but maybe in some of the cells, all right, you have a situation where the red doesn't show. You know, maybe this is like the pea plants, where if you're white, it's because you didn't make the enzyme, and if you're red, it's if you did. And maybe what happens is the amount of enzyme is different, so you get a mixing, right? Now, this doesn't really completely refute Mendel's work. Even some of the early investigators who worked on it may have thought that, because guess what? If you take two pink ones together, okay, like in the F2s. Now remember, I said early on in genetics, we thought it was mixing, tall parent, short parent, medium-sized kid, that kind of stuff. So it was kind of a mixing together. If you think about an art, if you made pink paint, you could do that, red and white, right? You couldn't take the pink paint and separate it back out into the red and the white again, could you? So if the information was truly blended in this genetic information, my analogy, pink flowers should make nothing but pink flowers. But if you notice down below, you don't get the phenotypic ratio 3:1. You don't get 3:1 because you only get a quarter of them are red, but you do get the genotypic ratio that we talked about. So you can look at this, and phenotype is now genotype, because the homozygous dominant ones are red. That's 1. Half of them are going to be heterozygote and be pink. That's 2. And then the other quarter will be white. Now, how do you know when this happens in the flower? Well, you don't by looking at it. You know it happens in a flower by doing it, by doing the cross, right? It's just a variation of Mendel's genetics. That's all. That's oftentimes what happens when people ignorantly discount established theories like evolution or even global climate change. These are not things that just came out of nowhere. These are things that have been looked at millions and millions and millions of times, and they've accounted for all the variations. Darwin's theory of evolution was correct straight out of the box back in 1859. The only thing we've done, though, is tweak and add to some of the things he didn't know about, because he didn't have the knowledge of paleontology that we have now, right? So when you hear about, oh, well, the scientific theory is incomplete, well, yeah, most of them always are. You know, the scientific theory of the rotation of the planets around the sun is incomplete, but it always explains it, because it always seems to happen. Okay, someone was asking me about eye color the other day, so I found this really cool chart. It's at the beginning of the chat. There it is. Let me click on that. Click on that link, that very first link. You guys see what I'm looking at? So when you click on the link, it should take you to a chart with some circles with O, A, B, and AB, some red circles. Now you see it? Yeah, Raymond, do you see it? Hey, Raymond, are you there? Okay, well, maybe having troubles with that, but anyway, we'll go on here. You refer back to that chart. Let's go back to the go back on into the class now and take a look at what I'm showing you. But you can use that chart in another tab to kind of refer back and forth. We talk a lot of times in human blood typing about this thing of the universal donor and the universal acceptor. That's what that chart shows you. But let me show you the basis of that. It is genetic. You see, we've been talking all along now about things that only have 2 alleles, yellow and green. Round and wrinkled, purple and white, unattached and attached, trotter and pacer. Most genetic traits, as you can probably imagine, aren't that simple. Many of them are multiple alleles. So what we mean by that, and I talked about this in class a little bit, is while you only have 2 alleles of a particular trait, because you only had 2 parents, That particular trait among the human population, which is really where the evolution occurs, folks, that genetic trait may have many, many, many alleles. In this particular case, to make it very simple, we're going to talk about blood typing in humans. Now, not the positive negative part. That's the rhesus factor. It's also inherited genetically, but my discussion is just going to focus on the A, the B, the AB, and the O. The basis of all that has to do with these things that appear on the surface of the red blood cells. They're called antigens. Antigens, for any of you who have allergies, I'm back in class. I'm looking at something that says multiple alleles, figure 1411. Is that where you are too? Yeah, okay. I'm sorry. Yeah, so what I was saying about blood typing is blood typing has actually 3 alleles here across the top. And again, the abbreviations are done for reasons that we don't really need to discuss, but just on the fact that you know that when something is in capital, it's going to be dominant. And right away, you look and say, huh, there's some interesting thing going on there. We got 3 alleles, number 1, instead of 2. And it appears that the A and the B are both dominant, and they are. This is an example, and I wrote down below in the red, of something called codominance. So we got dominance, we got incomplete dominance, and we have codominance. So the basis of being a universal donor or universal acceptor depends on whether you have these things called antigens. I typed it in before. An antigen is something that causes an allergic response. Antibodies respond to antigens. Okay, if you have allergies or have suffered from allergies, you know exactly what I'm talking about. Antigens can be pollen, mold, right? That could be an antigen. That stimulates your body to have an immune response. And part of that immune response would be the production of antibodies. And antibodies do exactly what they say. They are antibodies. They destroy things. They kill things. They kill foreign things in your body, things that aren't supposed to be there. All right, so let's look down below and look at genotype and phenotype and see how that affects this donating and accepting of blood. If you're homozygous dominant, or if you're heterozygous for the A allele, that means you have the A carbohydrate antigen, the little triangle, right? That means your red blood cells have an antigen on their surface that has a shape to it. And we're going to call it the A antigen. It's probably not shaped like a triangle because Mother Nature never makes regular shapes like that necessarily, but it does have a shape. The same thing with the B, but in this case, it's a different carbohydrate. Now, if you're thinking like carbohydrates we eat, no, remember on the surface of cells, there were glycolipids and glycoproteins. There were also saccharides that were, or not glyco, but there were sugars on the surface as well that identified that cell. It wasn't just glycolipids, glycoproteins, or also the polysaccharides. That's what I'm trying to say, the lipopolysaccharides. All right, so we have these carbohydrates sticking out. Says I'm an A cell, I'm a B cell. Okay, now with the codominance, if you happen to have one allele of each, you actually have both antigens. You have the A and the B, right? And if you're recessive, guess what? You have no antigens. It makes sense, right? Now, remember what I said. Antigens cause antibodies. So if you look at the case of the A, or if you look back, or if you have that chart again, you know what I'll do? I can just kind of recreate here, so you have to keep switching back and forth. It was O. What we say is O is a universal donor. O has no antigens. It won't elicit an immune response in anybody, right? That's why it is the universal donor. So this is the universal donor. O can donate to anybody. If you have A antibodies, if you have B antibodies, because guess what? If you have the A antigen, you have B antibodies. And if you have the B antigen, you have A Okay, hello. I'm sorry. I'm still recording. Yes. Sorry about that. Jeez. All right. So as I was saying, if you have the A blood, then you have antibodies against B, and the opposite for if you have B blood. And if you have A and B, you don't have any antibodies at all, right? You're not going to have any antibodies if you're AB because you have A antibodies that's going to attack your red blood cells, and if you have B antibodies, they're going to attack your red blood cells. That's why we say that AB is the universal acceptor. See my chart above? AB can take all the blood because it has no antibodies. O can be the universal donor because it has no antigens. So that's where the basis of all that comes. If you don't produce antibodies, then you're not going to have an immune response. If you don't produce antigens, then you're not going to elicit an immune response. But notice, like I said, this is all inherited through the same way with Mendel's work. You would do the same thing for each of these organisms up here. You would go and figure out what the gametes are going to look like as it comes to these alleles. So you know that half of the sperm will have the dominant A allele and the other half will have the dominant. Where it would be changed would be in these folks right here, wouldn't it? As well as the heterozygotes. Now, being homozygous recessive for a trait doesn't mean you're in the least proportion. For example, most people, I think most males, most Caucasian males, if I remember right, have about 47% or 50% have type O blood. There's some interesting things about where these different blood types came from. It's just some very cool things about human evolution and change through time. So, but it's outside the course here. So, any questions on that? O is the universal donor, but cannot accept. Right. O doesn't elicit an immune response. Yeah. All right. Now, this is an example of something called pleiotropy, sickle cell anemia. Pleiotropy is simply defined as one gene that controls many traits. So if you have the sickle cell anemia gene, if you have two copies, if you're homozygous recessive, then the hemoglobin is going to be sickle-shaped, and you're going to get all these problems you see down below in the green and the purple. Pains and fever, accumulation of sickle cells in the spleen, causing your spleen to rupture, the red blood cells break down. Traits don't have to be good, right? These are not good traits, but these are indeed traits. So if you're pleiotropic, you would inherit one gene and to control many traits. Now we're going to talk about polygenic in just a second, and polygenic is the opposite. Pleiotropic is one gene, many traits. Polygenic is many genes controlling one trait. But let's stick with pleiotropy for a second because there is something interesting that happens with pleiotropy. Sometimes in pleiotropy, what you do is you get a gene called an epistatic gene. The process known as epistasis. Now, the word literally means above the state or beyond. You'll see what I mean in just a second. Let's look at Labradors, black labs, chocolate labs, golden labs, all good dogs, right? Wonderful dogs. Let's look at the way it's inherited in them. The color of the fur is a dihybrid trait. So we've talked about that already, right? You see how that works. But there is something different about the E trait, okay? In the case of the E trait, if you're homozygous dominant, then the E trait acts as an epistatic gene. What we mean by that is the E trait tells the cells in the body of the organism, ignore what the other gene is saying. So the E, if you're homozygous recessive, like in the golden labs, notice the golden labs in the lower right. Some of them are homozygous dominant, which means they ought to be black. Some of them are heterozygous, which means they ought to be black. Some of them are homozygous recessive, which means they ought to be chocolate. But no. They have an epistatic gene. And if they're homozygous recessive for the E gene, then that says ignore or blank or mask is the word. Mask the expression of the other gene. Now, it can do this with one gene or many genes. People with albinism, right, tend to have the light-colored skin, eyes, and hair. That's all a bunch of different genes, but it's because they inherited the albinism epistatic gene. I like to think of epistatic genes as doing something in math, some big long equation, you know, a squared, blah, 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 all this crap in here, times 0. Everybody knows what that equals, right? That's always the easiest one on the test. No matter what's in the bracket, if you multiply it by 0, it's still 0, isn't it? So think about an epistatic gene as being multiplying it all by 0. So you might have several genes in here, right? This is a case of only one, but there might be tons in there. And they say, oh, you're going to have this skin color or hair color, all those variants that we talked about until you get that epistatic gene. Now, genes like that, especially ones that are epistatic, are usually detrimental to the critter. Like, for example, if you're a predator, let's say you're not predator, let's say if you're the white buffalo, he's at a distinctive disadvantage, isn't he? Predators can see him a mile away. Sharks have the opposite problem, by the way. There are instances of albino sharks that are born, and they have trouble hiding to stalk up on prey or even sneaking up on prey. Now, as you can probably imagine, all the way back from our discussion of metabolism, nothing's really easy as far as even in genetics, so that most of the genetic traits that we inherited, many of them are polygenic. And polygenic traits are easy to figure out in a population of organisms because if you plot the frequency, they make this bell curve. This is a picture from, I think, World War I, the average height of soldiers. For no other reason than just to take the picture, I guess. But, you know, there's some people that are on their short end and some people that are on the tall end, but most of us are about average height. These are traits that are polygenic, many genes controlling one trait, human height. Many traits are that way. That's why I said the other day, you could probably not find two people on the planet with exactly the same skin color, right? Take a look at this. This is just a real simple example. The colors represent whether you have the dominant or recessive allele. I don't expect you to be able to work one of these, but notice what's going on. And the color of the block down in the Punnett square is, let's say, skin color. So from very light colored skin to very dark colored skin. And this is only dealing with 3 genes, each of them having 2 alleles. Let's say it was 40 genes, each of them having 12 alleles. Right? Can you imagine doing the math on that? This is where computers come in to help, right? But this is how we get those variants of genes. Yeah, absolutely. You know, you could even have a situation where gene A has 15 alleles, gene B has 8 alleles, and a couple of them are co-dominant. You could add gene C and it's got, let's say, 28 alleles, and some of them are incompletely dominant. So you flip all them switches, man, and you come up with the incredible diversity. Thank goodness. The world all being the same would be so friggin' boring. Even if we look at flowers. Love flowers. Hydrangeas. These are hydrangeas. One of the things that we have found out as we go on through life in our study of genetics is that old nature versus nurture. In other words, are you the way you are because of your genes or have something to do with the environment? It's actually probably more a matter of what's down in the red, what we call our multifactorial traits, which is the genes plus the environment. So genetics provides perhaps a genetic blueprint, and then the environment modifies that. This may be the case in some, maybe not all genetic traits, but like in the case of hydrangeas, if you grow hydrangeas, you know that the environment, the pH specifically of the soil, the acidity or alkalinity of the soil determines the color. That acidic soil will produce blue flowers, and more basic soil will produce the red flowers or the pinkish flowers. Folks who want to make blue flowers put coffee grounds on them. Coffee is acidic. And if you're out there looking at your hydrangeas, my wife has tons of these things, and they're different colors, it represents the minor little differences of pH in the soil. It's really cool. It's like a real sophisticated pH probe. I'll go out and look at my wife's bush of hydrangeas. Like on one end, they'll be more red. The other end will be a little more blue. And I know that the pH is a little different on each side. It's very cool. So I think the answer would be to me on many genetic traits, maybe not all, I don't know, but many genetic traits, that there is a gene plus the environmental factor, you know? Okay, let's do this thing called pedigree analysis. This is fun because it involves detective work, right? Here's what we're looking at. Here's the question down below. Widow's peak. Widow's peak is back to being a real simple trait. You either have it or you don't. There's no or polygenic. There's none of that stuff in there. It's very simple, back to the purple and white flowers. But a pedigree, if you're looking at this, if you've not ever looked at a pedigree, they're really pretty simple. It's the family history. The males being indicated with the squares and the females as circles. And those individuals who have a trait, who show a trait, okay, or who have the phenotype, are going to be called affected. Affected because of you either have it or you don't. Not affected in the sense that there's something bad. It could be something bad, but whatever affected is you have the trait. You either are homozygous or heterozygous for the trait. And the lines between are the mating, and the stuff down below is the offspring. It's actually supposed to be a line right here, I think, to indicate the offspring. Firstborn on the left, secondborn, you get the idea, right? And then, of course, what's going on here is two families coming together, right? Male and a female from two different families. Okay, the trick is we're trying to figure out whether widow's peak is dominant or recessive. And the individuals that have the purple, they're the ones that have the widow's peak, and the ones in the white are not affected. The ones without the widow's peak, both males and females can get widow's peak. So the trick is, how do we figure out whether this thing is dominant or recessive? Well, if you look into there, we could try all these different crosses, but what I always like to do is I always look for what I call a telltale sign. I always like to look for a cross that's impossible. In other words, if I assume something, then this cross cannot happen. Now, if you're trying to think where I'm getting to, it's the same chart on the next slide. Look right there in the middle. Right there, what I've circled, what I put a box around in red indicates to me that two affected individuals, an affected male and an affected female, produced an affected female and a female who was not affected. Now, remember, there's a rule, very simple. Two dominants could make a recessive, but two recessives can never make a dominant, can they? Two individuals for whatever trait, I don't care, I'm just going to say A, my pen writes. Two individuals that are both homozygous recessive will never produce an offspring. You can mate 1,000 million times, never produce an offspring that is dominant whatsoever, will they? You can't produce an offspring with an allele that you ain't got. Makes perfect sense. So right there, what I put around the box must indicate that those two that are purple, they can't be recessives, okay, because they're the affected males. They have to be at least homozygous or heterozygous. I meant to say they must be at least heterozygous, right? See what I've done? I filled it in there. They have to have the capital W. They're affected. That is the only way they could produce children of each type. The alternative is if this trait was recessive, then these two individuals here could only have recessive children, and they don't. They have a mixture. So now when we have the chart like that, guess what we can do? We can go in and fill in the rest. Isn't this cool? Just one little bit of information. See, we can fill all this in. I'll go back. How do we know this guy is heterozygous, not homozygous dominant? Well, Tim said it much earlier. If this guy was homozygous dominant, this purple one right here, then you would never get recessive offspring, and he did. So that means he's heterozygous. Same sort of logic for all the other ones too. So try those out. They're actually kind of fun. But again, where to start is, and sometimes notice you can't answer everything completely because you don't have any information about this female's offspring. But look for those spots where things that are clear. And usually it's look for a spot where if it's impossible for two recessives to have children of mixed, right? As far as one having the phenotype and one not. Oh, good. I think I'm probably gonna. Where are we as far as the schedule? I think we're good as far as the schedule goes. Monday, Tuesday. Oh yeah, sweet. We're doing very nicely. Okay, Raymond, please what? I'm sorry, I didn't see the first part. No? I was talking about albinism a while back. Albinism is an epistatic gene. So in this case, we're going to call the epistatic gene A. All right. Now, again, we're going to be talking about albinism in terms of skin and hair color. Look at the picture of the two girls. They're obviously sisters, but one is an albino. Oh, you mean here? Let's go through it again then, really. Because this can be confusing at first, but then you go, oh, there is a real simple trick. So let's look at this chart. We get two assumptions. Two purples will only make purples, right? Two dominant individuals will only make dominant individuals, okay? And two recessives can only make dominants. So somewhere there's a violation of one of those assumptions there, right? If we look right here, if we say, okay, when an individual is affected, it doesn't mean it's dominant. It just means you have the trait. Remember, we're trying to figure out whether it's dominant or not. Two recessives cannot make a dominant. Two recessives cannot make a dominant. So right there, when you look at that part that I have in the box, that's going to tell you right away whether that trait is dominant or recessive. If we said that the trait, if the affected, being affected means you're homozygous recessive, that cannot happen. So it's not. It's got to be homozygous. It's got to be dominant, excuse me. Whether you have two alleles or one allele, you're going to have the dominant phenotype. And that means the trait is dominant. Try them a couple times. It's like you practice them a couple times and you go, okay, I get it. Yeah. All right, so these two girls are obviously sisters, but one is an albino. So the one on the left and the right, are presumably from the same parents, inherited all the genes about skin and eye color, or hair color, and even eye color, which is actually, I found out 2 genes now, not just one. But all of those polygenic multiple allele things came through on the girl on the left. They also came through with the girl on the right. But unfortunately, back to just simple Mendelian genetics, if you take a look, what she ended up getting is 2 recessive alleles. See, albinism is recessive. Many recessively inherited things or disorders are indeed that. They are recessive. For reasons that have to do with evolution, really. It's not the kindness of Mother Nature. Folks that have recessive alleles or homozygous recessive, if that trait is detrimental, they usually don't live as long or produce as many offspring. But notice something interesting about albinism. So you can have all these other genes. You could have B through J, and then you have A being albinism. Notice that if your homozygous dominant, you're normal, and that's fine. There's no big deal. That's going to be most of us, right? Most of us mate like this. But if you're a carrier, you're not going to express the disease, but that's where albinism is going to come from. So presumably, this girl's parents were normal in skin color, but they were carriers for it, weren't they? And if you do the probability of it, if they were carriers, here they are, they had a 1/4 chance of having an albino child. So if they had 4 children, probability, did it say that 4th one would be? No, but they said that the probability says that it should be. You can also say half of the offspring are going to be carriers, though. And then the 1/4 of them will be normal. So there are reports, documented reports of 2 African-American parents, 2 parents of African-American descent, having an albino child. You see how that can happen, don't you? You don't have to go through and change all the genes that make that individual its skin color it is. All you have to do is that individual has to be an albino. They have to be the product of 2 carriers. Amazing, isn't it? Well, albino. Albino, yep. No, albinism is the lack of skin melanin. That stuff that makes you brown when you lay out and get a tan, right? Folks of African descent or darker skin color are going to have more melanin, where those of the white and that are going to have less melanin. Yeah, I see where you disconnected for a second and came back, but it's about melanin that makes your skin color. So albinism is a lack of melanin. Folks with lighter skin color have less melanin. Are the rest of you hearing me okay, though? I can see where Beth and Lindsay are having troubles. Heck yeah, it is. Yeah, look, there are examples of albinism in the wild. Check it out, like the lions, but look at the guy, an individual on the right. Yeah. It still says it's being recorded. I hope it's not going to screw me on that. So albinism can occur. Yeah. Yeah, animals, and they do have a distinct advantage. Very hard to be a predator when you stand out like a sore thumb like this fellow on the right. Plants can be that way too. Look at that fern. That's an albinistic fern. Very distinct disadvantage, right? Can't photosynthesize. White gator's cool to look at, but man, hard to sneak up on stuff. This is a bamboo shark. White spotted bamboo shark. That's what they normally look like. I wrote a paper on this guy. He was a the first case in that species of an albinistic one. This was at SeaWorld. You can see the bottom that they usually find on, would lay on. These are lay-in-wait predators. Lay on the bottom until someone gets too close. A guy sticking out like a sore thumb is not going to be able to do it. Meter, or 3 feet, biggest. They never got any bigger than that. They lay eggs, actually. They're egg-laying sharks. Real popular in aquariums. Nobody, I bet. Anybody know who this is? He was an albinistic, interesting, William Nelson. Really bad day. Nah, it does look a little like him. Yeah, it does. What was interesting about these guys, this was a rock star back when I was a kid, late 60s, early 70s. This is, holy crud, I just drew a blank. But anyway, he had not only was he albino, but his twin brother, identical twin brother, was also albino. They did the song Frankenstein. It's on my iPad or my phone. Oh gosh, I'm drawing a blank. Edgar and Johnny Winter. Edgar and Johnny Winter. I don't know which one this is, but they were twins, identical twin albinos. So it was obviously part of their shtick. But Willie Nelson, that's funny. I like that. Oh, sickle cell anemia. Again, sickle cell anemia has to do with having the alleles, right? And what we're learning about it, yeah, Winter. Wow, I never thought about that. That couldn't have been their real name. Maybe that was part of the shtick too. What we're learning is you can be a carrier for sickle cell anemia down here. And unlike albinism, where if you're a carrier, carriers for albinism look they're normal. Their skin isn't a little bit lighter, right? They're just very normal. But if you're a carrier for sickle cell anemia, what we're finding out, and Ryan Clark from the Pittsburgh Steelers almost found out in a wrong way. He was playing for the Steelers at the time, and they were having a game in Denver, which is obviously higher in altitude where there are low O2 levels. And I believe he knew he was a carrier, but I think the assumption back then, if you were a carrier, wasn't a problem. We'll come to find out, or else he didn't know he was a carrier. I don't recall the story exactly. But he had to go to the hospital. He almost died because of the very low O2 levels up there in Denver. His carrierness came into play, and actually some of his red blood cells that were normal in shape began to actually shift, right? And actually become sickle shaped. Remember, though, I said it's interesting. Why are there still carriers around? It's actually a Bio 2 question, but when you're a carrier, you don't succumb to malaria as bad as other individuals. You're not immune to it, but you survive it a lot better. Now, some traits which are dominant, I don't want you to confuse being dominant with being very common. Like, for example, some traits are dominant, but they're not common. Like the one that causes achondroplasia. Achondroplasia. I always got to remember that means without a part of the bone forming. Folks with achondroplasia, holy cow, are short. They're dwarf is the name we use for it. And basically the way you get it is if you're heterozygous for it, okay, so that's the dominant allele. If you have at least one dominant allele, you will have this condition, this achondroplasia. That in order to be normal, which most of us are, or without the condition, you're going to be actually homozygous recessive. Again, remember I told you that half the population has O-type blood, even though it's recessive. In this case, you got to, chance-wise, you got a better chance of having the dwarf allele. Why are there not more folks out there suffering from achondroplasia? Well, the gene is inherited this way, but the gene is not common. You understand what I'm saying? Remember, the gene is the spot on the chromosome. So some of us, many of us, most of us don't have that gene. But if your mom and dad did happen to have that gene, right? So first of all, there's the probability of that. And then the probability down below, half of it, is that you would end up with the condition. So the doctor over here, right? You have normal life otherwise. You just shorter than normal, right? Now, these kinds of genetic disorders, my last slide, good, because I have one more minute. These kinds of genetic disorders obviously can be determined after birth, but even prior to birth with processes that you heard about, is at least the amniocentesis. The amniocentesis, they do about week 14 through 16, where they inject a needle actually into the amniotic fluid. The amniotic fluid is the fluid that surrounds the baby. When a woman's water breaks right before delivery, The fluid, the water that comes out, is the amniotic fluid around the baby. Now, just as kind of a neat sideline, I know we're at time. If you give me just one more minute, having the fluid around the offspring, having that amniotic fluid, is what allowed us mammals and us reptiles to move away from the ocean. But whole different story. Again, that's bio 2. But they can draw fluid from there, or they can do what's called a CVS, is the chorionic villus. Chorionic villus is the part of the placenta. Villus means fingers. It's the interweaving of the placenta with the mother, so that they can transport the nutrients. But anyway, the chorionic villus is all baby DNA. It's very concentrated. The amniotic fluid is not so concentrated, and that's why it takes longer. Amniotic fluid, it takes a few weeks, whereas you can do it a few hours with CVS, which is done early on too, 8 to 10 weeks. And what you do is you do a karyotype. You draw out some of the cells of the baby. That's why you do it with CVS. It works quicker because you get more. You draw it out and do karyotypes, and you can look at genetic disorders. Not only numbers of chromosomes, but they can also tell whether genes are absence or presence in some cases by doing these biochemical and these genetic tests. Cool stuff, man. Cool stuff. Okay, let me stop the recording there.